Hey everyone, and welcome to my world. I'm a mycologist working in a hospital lab, and honestly, my job is a lot like being a detective. Every single day, I have to figure out which organisms are the real fungal culprits, and which ones are just very, very convincing imposters. So let's dive into this hidden world of biological lookalikes. This is the one question that's on my mind with every culture plate I look at. You'd think that in a nice, controlled lab, telling things apart would be easy. But it turns out some of the most convincing fungi we grow from patient samples, well, they aren't fungi at all. They're imposters, hiding in plain sight. So before we can spot an imposter, we need to know the rules of the club. To get into the official fungus club, an organism needs a few key things. A cell wall made of something called chitin. It has to eat by absorbing nutrients from its surroundings, and it can't have any chlorophyll. These are our ground rules, our field guide for identifying the real deal. Okay, so our first group of suspects are bacteria. These guys are the absolute masters of disguise. See, they're prokaryotes, which means their cells are way simpler than fungal cells. They're in a totally different domain of life. And yet some of them have evolved to look and act so much like the fungi we study. It's a classic case of what we call convergent evolution. Basically, unrelated organisms end up looking alike because they've adapted to similar lifestyles.
All right, meet the Actinomyces. This is a huge, really diverse group of gram-positive bacteria, and they are, without a doubt, the most common fungal mimics we run into. They're the reason a lot of mycologists, myself included, have to be pretty good bacteriologists too.
So why are they so tricky? Well, for starters, they build these delicate, thread-like structures that look almost exactly like fungal hyphae. We call them false hyphae. And the real kicker? They grow slowly, just like a fungus. A normal bacterial culture is ready in 24, maybe 48 hours. But our fungal pleats, we incubate those for weeks. And these guys take their sweet time showing up right on our turf, growing beautifully on our special fungal media. On top of all that, they can cause infections that look just like fungal infections. So you can see why they really keep us on our toes. Next up, we have the mycobacteria. This group includes the infamous bug that causes tuberculosis. Now, if you look at them under a microscope, they're less of a fungus mimic. They don't really form those filaments. So you might be asking, why are they even on this list? Well, it comes down to a really important, very practical reason that shapes our daily life in the lab. These bacteria are incredibly tough. Their cell walls are packed with these waxy substances called mycolic acids, which makes them super hardy and, you guessed it, very, very slow growing. So just like the Actinomycetes, they love the long incubation times of our fungal cultures. But the most important thing we share with them isn't biological, it's logistical. They're dangerous to work with. And this is why we group them together. Both the really dangerous fungi and mycobacteria require what's called Biosafety Level 3, or BSL-3 precautions. That means we have to work in a special room with negative air pressure, so nothing nasty can escape. We're talking a full protective lab coat, gloves, and breathing through either an N95 mask or a big helmet called a PAPR. And because those safety requirements are identical, in most hospitals, the mycology and the mycobacteriology labs are actually the very same lab. All right, our next imposter might just be the most surprising of all. I mean, when you think of algae, you're probably picturing green stuff in a pond, right? Not something that can cause a human infection. But in the world of immunocompromised patients, pretty much anything is possible. I absolutely love this description. It's so vivid and perfect, because when you look at this thing under the microscope, that's exactly what you see. These perfect little round structures with even smaller spores inside, arranged just like the spokes of a bicycle tire or the segments of an orange slice. It's an image you just don't forget. So what is it? It's called Prototheca. It's technically an alga that, somewhere along its evolutionary path, lost its chlorophyll. It's adapted to a parasitic lifestyle, and it can cause a really rare but serious blood infection called algamia, almost exclusively in patients with severely weakened immune systems. And, of course, it grows perfectly on our fungal media. Okay, for our last group of imposters, we have what I like to call the kingdom hoppers. These are organisms that science has had a really tough time pinning down. Their classifications have actually changed over the years, which is a great reminder that our understanding of the tree of life is always, always evolving. And the absolute poster child for this has to be pneumocystis. This is a hugely important pathogen, famous for causing a really severe pneumonia, especially in HIV patients early in the epidemic. and for decades, scientists just could not agree on what it was. For most of its history, Plumocystis was classified as a protozoan, a type of parasite, mainly because its life cycle looked a lot like other parasites. But then, as our genetic sequencing technology got better and better, the data became undeniable. Its DNA is fungal. It's the perfect example of this flickering classification, an organism that was literally caught between two worlds. Next up, slime molds. I mean, look at them. They look so much like fungi. They make these little fruiting bodies and release spores. But when you look closer, the differences are huge. In its main feeding stage, it's just a giant single-celled amoeba. And most importantly, remember the #1 rule of the Fungus Club, a chitin cell wall. Well, slime molds use cellulose, like a plant. And that's a deal breaker. They also have a different life cycle than most fungi. It's just a totally different kind of organism.
And that same rule, the cell wall rule, applies to water molds. Their cell walls are also made of cellulose, not chitin, but it goes deeper. On evolutionary level, they're in a completely different supergroup. A big reason why is their spores have two different kinds of flagella for swimming. True fungi are opisthecons, which means rear flagellum. And get this, so are we. Human sperm cells have that same single rear flagellum. It means we're actually more closely related to fungi than water molds are.
So we've met bacteria and alga and these former fungi. But you might be wondering, why does sorting all of this out really matter outside of a biology textbook? Well, in a hospital, this isn't some academic exercise. It can literally be a matter of life and death. So here it is, our final lineup, the mycologist's rogues gallery. You've got your bacteria, the actinomycetes and mycobacteria given away by their false hyphae and waxy walls. You've got our rogue alga, Prototheca, with that unforgettable bicycle tire look. And our former fungi, the slime molds and water molds, betrayed by their cellulose cell walls. Each one has a tell, and it's our job to find it. And this is the bottom line. An antifungal drug will do absolutely nothing to treat a bacterial infection. The right way to treat an algae is different from how you treat a fungus. A correct identification leads directly to the correct treatment for the patient, period. And for me, on my side of the lab bench, knowing whether I'm handling a simple mold or a BSL-3 level bacterium determines exactly what I need to do to keep myself and my colleagues safe. So, we end right where we started, with a question. The lines we draw between the kingdoms of life are at the end of the day human inventions. And as our genetic and molecular tools get more and more powerful, those lines are going to keep shifting and blurring. It really makes you wonder, what other imposters are out there, just waiting in plain sight, ready for us to discover? Thanks for joining me in the lab today. Hey everyone, and welcome.